I'm Tamas Varga, and I'm an EMSL. I'm using X-ray techniques, including cyclotron techniques for environmental and biological problems. And this project is an example of that, where we used two other synchrotrons, and this project doesn't have a computation component, but hopefully it might generate some thoughts or even discussion for that kind of application. I'm going to tell you about endophyte-promoted phosphorus solubilization in poplar tree. And this has been a collaboration where we used synchrotron micro XRF coupled with XANES and also lab and synchrotron X-ray CT, as well as some lab techniques such as fluorescence microscopy, optical assay, and proteomics. So why do we care about endophytes? These bacteria are known to promote nutrient fixation and, for example, nitrogen or uptake through solubilization in plants. And so we wanted to provide evidence that select strain of endophytes is able to do that in poplar. And so basically, to achieve this goal, we had, in collaboration with Professor Sharon Doty at UW, had two group of samples. One group that didn't contain the bacteria was called control, and the other group, which was inoculated with two different strains of endophytes selected by the Doty group, was called PMIX group. And both sample groups were, the poplar plants were grown under the same conditions, in the same medium. They were kind of nutrient limited, and to both groups we added insoluble, non-water soluble tricalcium phosphate so that we can track solubilization. And we wanted to provide basically evidence of this endophytic activity, which I'm hoping that by the end of the talk I convince you that it happens, to try to visualize this endophytic phosphorus uptake and try to understand the phosphorus chemistry during solubilization. So after a few weeks of growth, all samples were harvested, and of course we measured the physical characteristics such as leaf mass and root mass, leaf weights. Although there was some indication that larger growth was achieved in the PMIX group, these results were not statistically significant enough to attribute larger growth to the endophytic activity. So we needed more evidence. So before moving on to the X-ray studies, we showed in the lab using colorimetric assays that the two select strains, WP5 and WP42, are indeed able to solubilize phosphates. And so as you can see on the left, both strains, see if I can find my pointer. Okay, so on the left, you can see evidence of significant solubilization from calcium phosphate compared to the control in both strains. And we also check for other important phosphates in the soil, such as aluminum phosphate and iron phosphate. Both strains showed some limited ability to solubilize the aluminum compound, and only one of them, the WP5 alone, showed some greater ability to solubilize that phosphate, iron phosphate, than the control. Next, we looked at whether these bacteria can colonize on the poplar root and in it. And so this is important because these kind of bacteria live in symbiosis with the plant. And so this fluorescence microscopy experiment was used to show that. On the left side, we are showing a root surface. So the two different strains were tagged with two different fluorescent proteins to visualize them. And what we can see here is that the bacteria indeed colonize on the root surface, creating kind of a biofilm, which suggests that the bacteria are able or are available on the root surface to manipulate external soil chemistry. And then the other figure or picture, I'm showing a lateral root section on the primary root. And here we can see that the bacteria are beyond the epidermis, so it's basically inside the root tissue, suggesting that they are available for the manipulation of the internal chemistry as well. Some of the roots were taken for CT imaging. On the left, these are whole roots that were imaged in the lab at EMSL, and we also imaged small sections at high resolution at the ALS 832 beamline micro-CT. And in all groups, what we found is that the PMIX samples had a greater root volume, while the controls had small volume and the largest root surface area, suggesting a lot of fine root growth. And so this suggests that there was something going on with the better availability of nutrients, possibly in the PMIX group. To gather more evidence of this endophytic activity, we went to do X-ray fluorescence mapping and Zane's spectroscopy at the XFM beamline at ALS, beamline 1032. This is a sample holder which shows that some larger roots mounted, but we also had created thin sections so that we can investigate the what's going inside the root, not just on the surface. Yes, and we did both to get an overall picture of the speciation elements. We did defocused measurements as well as microfocused mapping and Zanes to study the hotspots, local spots. So these are just some examples. So these measurements were done about 100 EV above the phosphorus K edge. So obviously we were able to capture phosphorus, but also some of the elements that were excited at that energy, such as calcium, potassium, aluminum, silicon, important in the soil. Calcium is especially important because it was present in the compound that was added to study solubilization. These are some examples of maps, grayscale images from the fine microfocus fluorescence mapping. This is for phosphorus, calcium, and potassium. You can see that there is some correlation between the phosphorus and the calcium, and this is just an example where we show that some of the spots, like hot spots and general spots, where the Zane spectra were also taken for further analysis. But this is the slide where I would summarize the micro-XRF data. Here we are comparing PMIX and control samples, and I like to bring your attention to the bottom two images, which are grayscale images brought to the same phosphorus intensity scale so that we can directly compare phosphorus distribution. And what I'm showing here is kind of general view among the samples, that while the control had this homogeneous phosphorus distribution in this longitudinal root section, and these are thin sections, the PMIX samples showed a lot of high concentration spots, hot spots containing phosphorus. So there was a clear difference between this, and this suggested that in addition to the phosphate, which we probably see from the Hoagland solution that was added to both samples, the nutrient mix containing ammonium phosphate, in addition to that, we have additional phosphorus coming from possibly the solubilization. So we wanted to look at those. Before I go there, I also wanted to report that using another beam time at the APS, Advanced Photon Source Beamline 2 IDE, we took couple dozen samples or locations to get a statistically relevant picture of the phosphorus concentrations between PMIX and control samples. So we did this extensive analysis, and we found that the phosphorus concentration was consistently greater in the PMIX samples by about 20-30% on average than in the controls, again indicating greater phosphorus uptake in the in that group. So we wanted to understand the chemistry of the phosphorus solubilization in these samples, and to that we did Zanes at beamline 1032 along with the microfluorescence. First, we did a bunch of standard compounds, collected standard spectra, so that we can later fit them in organic and inorganic. Inorganics were calcium phosphates and pyrophosphates, hepatite. For organics, we did DNA, phytate, lecithin, etc., and some other phosphates. And also, I'd like to thank to Peggy O'Day at Stanford, who also provided some of their library from their group phosphate library. And so using those standards, we fitted both the defocused Zanes and the focused microzanes to the standards. We used linear combination fitting to determine the phases and the relative amounts. And these are for the data, but I just like to summarize it here. So in general, we found that the chemical state of the phosphorus inside the root was a mix of organic and inorganic phosphates in both groups. The greater difference was shown in the hotspots, which have very different compositions. Specifically, the PMIX samples have a significant amount of inorganic phosphate, which were in the hotspots, and those corresponded to mostly calcium phosphates. And this is consistent with the assumption that the tricalcium phosphate was solubilized and then ended up inside the root in some form. And the interesting thing that those hotspots are inorganic calcium phosphates suggests that the solubilized phosphate might have reacted with cations like the calcium inside the root and became insoluble again. And this is something that we would like to study further, but it suggests that the endophytes might have the capacity to re-release phosphate inside the plants, as opposed to, for example, rhizobacteria. And also the organics identified were mostly phytates and DNA, and that was present in both groups. So after that, we also tried to look for further evidence of endophytic phosphorus solubilization using proteomics at EMSL. And this slide shows the KEGG functional category enrichment distribution. So this KEGG analysis basically looks at which proteins are most significantly changed or altered in the two groups, and then these are proteins related to different functions such as metabolism, genetic information processing, etc. And what we found that inconsistent with the greater access to phosphorus in the PMIX group, more significantly changed proteins were found in the PMIX group, more KEGG functional categories, and that suggests that the greater access to phosphorus stimulated more metabolic pathways. Also the
Tamas, this is also your 2-minute warning.
Yep, thank you. And the transporter proteins that were generally increased in the control, one of them, the ATPase, that plays a role in the cell replication that supports increased growth of root cells, showed increase in the PMIX roots. And in contrast, in the control samples, we saw enrichment of proteins involved in phosphorus sensing, photosynthesis antenna proteins, and proteins in general that suggested that oxidation stress, nutrient limitation was dominant in the control groups. And then finally, the string network analysis, which took the most changed proteins from the previous analysis, showed the importance of a specific protein. Centrality is basically the measure of importance here, and this UMP synthase protein, which is known to be interrupted if there is a nutrient limitation. It is known that plants tend to deliver sugar to the roots, and the disruption of this protein is related to the phosphorus availability, and so that should provide a further evidence that this endophytic activity is existent in the PMX group compared to the control. So just to summarize, we found that the poplar samples that contain the bacteria exhibited a greater growth of their root architecture, and the control samples exhibited an increased fine root growth. This from CT, and this is consistent with the nutrient limitation as the control was striving for nutrients. And we visualized the phosphorus inside the root by synchrotron micro-XRF, and that showed the capacity of the endophytes to solubilize phosphorus or phosphates. And Zanes suggested that while both groups contain a mix of inorganic and organic phosphates, the insoluble tricalcium phosphate must have been solubilized, and some of it was present inside the root for the PMX samples dominantly as inorganic hotspots. And an interesting finding that which suggests that these endophytes might have an advantage over rhizobacteria in that they might be able to re-release phosphates inside the root and not just outside in the soil. And proteomics provided further evidence of this endophytic solubilization. And with that, I'd like to thank my team. Professor Sharon Doty provided the basic guide. I contributed to her EMSL user project looking at poplar from the nitrogen fixation point of view, but be able to provide this project in parallel. And all these people are also co-authors on the paper that is shown here if you want to check it out for more details. And I have to thank for funding for this specific strategic science area call that started 2 years ago, and also BR and these Office of Science contracts for the synchrotron experiments. With that, I'd like to thank you for your attention.